So continuing on with some of the interesting presentations from the meeting, Richard Jopie from the University of Alabama, Birmingham, has picked up on the earlier work of Rob Bockwitz, and in fact has collaborated with him a great deal, looking at the therapeutic effects of lithium, especially in rescuing the signaling pathways that are dysfunctional in Fragile X. He's also looking a lot at GSK3 beta. It's a really important enzyme in regulating a lot of cellular functions, and it appears to be itself misregulated in Fragile X. So in other words, you don't have too much of it per se, but you do have too much activity of this important enzyme. And GSK actually stands for glycogen synthase kinase, which gives you a hint that it's involved in a lot of cellular processes, including energy metabolism, glucose metabolism. So GSK3 beta inhibitors are actually being developed for a number of indications by many different pharmaceutical companies. One of the potential indications is for the treatment of psychiatric disorders, but it's also being looked at as a potential target for a lot of other things. So far, nothing we've found is actually a better or more useful GSK3 beta inhibitor than lithium, which is already available and has already shown some at least preliminary evidence of therapeutic effects in Fragile X through Liz Berry-Kravis's excellent study. But the lab at UAB is continuing to look at some of the important aspects of GSK function in Fragile X, and they're doing some really good stuff. Hopefully we'll be expanding on that in the future. Len Kaczmarek from Yale also presented subsequently talking about his work on various ion channels in Fragile X. We tend to talk a lot about mGluR5 and different kinds of receptors, and then we talk about signaling pathways that are coupled to those receptors. But one thing you don't hear a lot about is ion channels. And it turns out that there are a number of interesting and previously unknown mechanisms for regulating the conductance of ion channels in neurons. And that's important because that has a direct effect on the electrical conductance in those neurons. So neurons are to some extent like little tiny computers, and they conduct electricity, and then they spew out chemical signals. Well, the electrical properties of the neuron can actually be directly regulated by not just altering the number of ion channels that the cell produces and inserts in the membrane, but even after those ion channels are in the cell membrane, they can be regulated. So they can be upregulated or downregulated. And Len and his fellow researchers at Yale have shown some very interesting effects in Fragile X. Specifically, FMRP, the missing protein, normally binds to one of these potassium channels called SLAC and actually regulates it by direct protein binding. And that's really important, and that's kind of a new mechanism in neurobiology. So this is something that's really on the cutting edge, and when I studied neurobiology 20 years ago, nobody said anything about this. I don't think they had the slightest clue this kind of thing was going on. It's a whole other level of complexity. But more importantly, there are SLAC inhibitors. So there are SLAC channel drugs that can affect the conductance of this channel. So there are drugs that you can use to increase the open states of these channels, so channel openers, and then there are channel blockers. And drug companies are working on these things, and they are investigating a number of diseases, and this concept of channelopathies has occurred. So certain people think that certain diseases might actually be the direct result of misfiring ion channels. And it turns out that if you look at genetic studies of autism, there are a number of genetic forms of autism that are attributable to mutations in genes for potassium channels. Now, I don't think any have been identified with SLAC so far, but another channel that's been identified as being downregulated in Fragile X is called BK or MaxiK, and there are BK channel openers that have been studied for a variety of indications, none that are on the market, and as far as I know, none that are in human trials right now. But this gives you an idea of how we can repurpose existing drugs to treat Fragile X. So we can approach drug companies and say, hey, you've got this drug in your vault and you're not doing anything with it. It doesn't work for the original indication that you were trying. We think it might actually be useful in Fragile X. Why don't we give it a try? And a lot of companies are receptive to this approach. Timing is everything. You have to catch them at just the right time. If it's something that they haven't been working on actively for many years, sometimes they're reluctant to dust it off or even try to retrieve it from their archives, because some of this information has been inactive for years, and the personnel that were actually working on the project are long gone and have moved on somewhere else. So you have to catch them at just the right time, but that's a lot of what we spend our time doing, is trying to collaborate with these companies to see how we might be able to repurpose some of these drugs. So Len is working on some drugs that have effects on SLAC to see if they might actually be able to ameliorate these phenotypes. He's a kind of an interesting guy who is a musician himself as well as a neurobiologist, and so they're looking in auditory cortex, not surprisingly. And it turns out that a lot of these potassium channels require ultra fine tuning for a number of auditory functions. For example, if you want to be able to detect the tiny differences in frequency and amplitude that allow you to tell which direction a sound is coming from, then you need to be able to match the signals very closely. And so these potassium channels and the ability to upregulate them and downregulate them turns out to be incredibly important. Well, after those presentations, which I think everybody found really, really interesting and exciting, we moved on to something that is maybe even more interesting and exciting, which is clinical trials in Fragile X. We decided not to do the usual thing and hold those till the end of the meeting when everybody's trying to leave early. But we decided to do some clinical trial talks early on in the course of the meeting so that people could understand where all this was going. I think it helps to give the basic scientists a little perspective about what it is we're trying to do with all this research. So Craig Erickson, who's at the Indiana University School of Medicine, one of the best academic psychiatry clinical trial sites around, has been doing a number of small pilot trials looking for potential available agents that might be useful in Fragile X. Presented his results, where he had very strong results with aripiprazole, otherwise known as Abilify. Most of us parents know that Abilify is a really good treatment for Fragile X, but this is the first actual clinical trials data that we have that we can point to that says, yep, this is good. And not only that, but the open trial that he did will lead into inclusion of a Fragile X arm of a much larger autism trial of aripiprazole. So they'll officially get an indication for aripiprazole for autism, most likely the same as they have for Risperdal. And I'm not sure that they'll actually get a specific indication for Fragile X. I doubt we'll have enough patients to make it significant, but there will be a double-blind, placebo-controlled follow-up. And so you'll be able to reinforce these findings with some controlled studies. And then the folks from Seaside Therapeutics actually presented some of the initial analysis of a fairly large phase 2 trial of baclofen, or rather R-baclofen, which is their proprietary version of baclofen that they're developing as a therapeutic for Fragile X and autism.